I'm recording now. If you're watching the video, it's going to be business as usual. There's going to be a series of attendance words that you're going to send me, email me, to get your points. Every time I say an attendance word, every time, not just the beginning, but every time I say an attendance word, you'll need to take a screenshot. And the last word for attendance is going to be me asking you to ask me a question about the material we just covered. So as you're listening to the lecture, keep that in mind. We'll be ready to ask a question at the end. It is about 7:33 now, so you can skip ahead about, let's say, 25 minutes if you're watching the video. Huh. That looks odd. What? What in the world? Okay. Interesting. Whatever.
There's people inside this time.
Yeah. Yep. Mondays.
Yeah.
So are you on the travel team, or did you, so you got a relaxing weekend over at least?
For the most part. But the way Coach Flynn is in, I think everybody's going to trip. Which, I mean, which I think is the right thing anyways. Because I was like, all right, I did my part. I raised enough money for my part to I should be able to get.
Yeah,
Like,
that's only fair. Yeah, they're selling tickets for people like fans who want to go.
yeah, for sitting on the plane, but
Yeah.
because there's a whole form, and it's like you're responsible for your hotel, like transportation anywhere. It's like make sure you're at the airport.
Yeah.
Because we're leaving next Friday morning. And it's like we get down there, and on our schedule it says beach trip, and then like stadium walk through.
How long's the beach trip?
Pretty sure it's 2 hours.
That's cool.
Oh, there's the beach, we gotta leave.
Yeah. It's a nice beach though. It's really white powdery sand in there.
See, I ain't never been to the beach in Florida. We always just went to like Oak or
Morning.
Morning.
Well, those two places would be more like the east coast of Florida, has that kind of beach. Daytona Beach, St. Augustine Beach, stuff like that. Miami a little bit. But when you're on the Panhandle and the Gulf, which is where you guys are going, it's, in my opinion, much nicer.
It's like, my grandma used to say, "I think that's the way." Like, "Oh, this is really at home." That's what. She's like, "That's good for kids," but look, the mom said, "Mama, it's less work. That's what it's gonna be."
Yeah. Damn near in Alabama, it's in the other time zone.
Yeah.
That was my favorite school I went to in Marines. It's like 6 months there. End of the time, my mom had just moved back to Tallahassee. So it's like right down the road. When you're 18, you really miss your mom. So it's nice to be able to see her. morning. All right, Brumfeld is not here yet. Champion is here. Gross not here. Gunther is here. Mr. Hill is here first. A little extra credit for that. The other Mr. Hill is not here yet. Jackson, not here yet. Keys, not yet. Perez is here. Sigmund, not yet. Slaughter is here. Smith is not here yet. Stover is here. Tompkins is not here yet. The Nest is not here yet. Watson, not here yet. Young is here. Zweiback is here. Lawrence is here. Nope, Lawrence is not here. Sorry. Oh, yeah. Lawrence is here. Wilson's not here yet. Smith just came in. Have I missed anybody? All right. We still have about 2 minutes. So I'll do the repetitive announcements. Because again, I've been saying this every day. So no one, if somebody comes in at 8 and they missed it, they're not really missing anything. Attendance is mandatory. I've turned it into the university. Participation is graded. If you're here today, you do not have to send the words in, but you can for extra credit, as long as you get them in before 9 a.m. at the end of the lecture. If you're online, as usual, you write down all the attendance words, but you also say a screenshot or take a screenshot. Again, if you're watching the video, that is, if you're watching the video, take a screenshot every time I say the keywords, and at the end of it, email me the list of words and the list of screenshots. And the last word for attendance, if you want to call it a word, is going to be you asking me a question about the material presented in today's lecture. So any questions about attendance? Same stuff, right? Nothing new. But again, even though I don't have it written on here, I'll remind you again, since we're finally starting to get into some cooler weather, make sure you check your emails, of course, announcements before you come to campus, especially if it's like if you don't live on campus and it's a whole thing for you to get here, you should definitely make sure that I have to cancel it or the school has to cancel it. So yeah, before you come in, make sure you check your emails, of course, announcements, make sure we have a canceled class. Or I guess a more accurate way to say that would be moved class online, because unless something happens to me, it's probably not going to get canceled. And even then, I might find somebody else to do the lecture for me, because you guys paid for an education, so we'll get it to you. Anyway, saw some people come in. Let's see here. Tompkins came in. Sigmund too, right? Yep. Tompkins and Sigmund came in. Anyway, other announcements. Like I've been saying, midterms are now past us. Sunday was midterm, so I'm turning in those grades as soon as I can, hopefully today, tomorrow, or the latest. And to remind you, there's nothing to worry about. Right? You could have an F right now in the course with an A, and according to your transcripts, anybody who looks at your transcripts, it's like there's no difference, right? It's what you in the class with. But on that note too, I will say historically, usually what people have in midterms is what they end up with. So, but that is up to you. That can be a good thing or bad thing. So if you have a good grade right now, you don't want to just rest on your laurels and be like, I can slack off a little bit now. Don't do that because then your grade will go down. But like I said, historically people who have those grades keep them. But that is up to you. So if you have a bad grade right now, you can still fix it. You still have half of the semester to fix it, and I'm here to help you fix it. You can take care of late work and fix old mistakes. Watch the videos of the lectures for the days you missed, so we can get you caught up. But now's the time to do it, right? We're halfway through, so let me help you. Any questions about midterm grades? All right, it's 8:01. Wilson's here now. We'll get us started. Get us officially started, that is. So, it is now too late to retake exam 1. Just as a reminder, if you did retake it, you need to let me know, because I have no way of knowing that I need to adjust your grade if you did. So now that it's too late to retake exam 1, you can meet me online. Well, I guess you can meet me in office hours too. But anyway, you can meet me, and we can go through your exam and talk about your incorrect answers. Like, why was that incorrect? Then I give you the correct answers and talk about why this is correct. You'll get a 5% boost to your grade for doing that. And also, remember, your final exam, about half the questions, about 50 questions, are going to come directly from your first 3 exams. So your first 3 exams are your study guide, partially, for your final exam. So meeting me to get all the correct answers will be very beneficial for you. Like I said, you get your 5% boost, and you'll get all the correct answers. So any questions about exam 1? All right, it is now too late to take exam 2. It closes on Sunday. I reminded everybody a lot. I hope nobody failed to take it. I haven't looked yet. So I will reopen it today or tomorrow so you can retake it. It'll be just like the first one. The retake's going to be open book, not timed. You can retake it as many times as you want. What'll happen is you'll let me know when you're done retaking it, and I'll take your highest grade and your first grade, and I'll combine, I'll average the 2. So even if you missed taking the exam 2, you can still take it. Of course, your first grade is a 0. So if you retake it, then even if you get 100, your average will be 50, but that's still better than a 0. So anyway, any questions about exam 2? All right, that means exam 3 is now due in 4 weeks. And this is stuff we've been working on, right? Basically reproduction. So speaking of which, we finished with cellular reproduction, the 2 different chapters of that. That's what we finished recently. And then today we're going to go into genetics, which is basically part of reproduction. So all that will be on exam 3 in 4 weeks. So no questions about that. All right, pre-lab was due on Friday. It's way past due. That's the bad news. The good news is I've been really busy because it's midterms for you guys, but it's finals for my other online course, one of my online courses. So I have to prioritize them. So I've been grading a lot of their stuff and helping them along. Therefore, if you haven't turned in the pre-lab yet, I don't know. Now, granted, when I go to grade it, it'll tell me when you turned it in. But I don't care. If you haven't turned it in yet, go ahead and get it in. Because, you might lose some points for it being late, but at least you'll get it, right? At least you'll get it graded. So as long as it's turned in before I get to grading it, I will grade it. If it's after that, you just get the goose egg. No one wants that. So anyway, any questions about the pre-lab from last week? All right. We will meet in person as usual for lab this week. Wednesday, 10 a.m. Yeah. Don't need a computer or anything this week. Next week, it's another virtual lab. And it doesn't really have a pre-lab. So, hey, there's good news, right? There's no pre-lab that you have to deal with this week. Also, because it's a virtual lab, maybe it'll be like we did last week. If enough people get it done, then we just won't, it'll be non-mandatory for you to be here. Because again, it's virtual. But yeah, I'll let you know about that. That will depend on how you guys did last week. Because again, I haven't rated your virtual labs from last week yet. So if they were really great for the most part, then we'll just keep doing what we're doing. But if they were horrible, then we'll have to make some changes. So anyway, any questions about the lack of pre-lab or the lab that's next week? All right, do you guys have any announcements or reminders or questions? Nice and easy. Hopefully you guys talk more. Like I said at the beginning of the semester, I don't want to talk at you. I'd rather talk with you, have some conversations. But anyway, let's jump into it. We are in a new chapter. Why genetics? Well, first of all, this chapter is about genetics. I think your textbook calls it, well, I don't know, I'll get there. I have it written down somewhere. I forget what they call it, but it's basically genetics, right? And I'm sure you've noticed the pattern here, just like the past few chapters, I'm basing this lecture off of the old textbook because I think the old textbook did a better job of presenting the information. So I'm going to present it the way the old textbook did. The information is the same, but the way I'm presenting it is different. But I'm only telling you that because, again, if you're like one of those students who likes to follow along with the textbook as I'm teaching, it's going to be in a different order and there's going to be different examples. So I just want to make you aware of that. Anyway, the old textbook always starts with these pictures and it poses the question like, hey, why does this chapter matter? So I guess we're a little bit ahead of the game. Might as well take some time with this. So what color, how would you describe the color of that woman's hair? Someone said, hey, what color is her hair? What would you guys say?
Brown.
I would say that too. My wife probably just give me some kind of technical term. It's Auburn number 5 or something. But yeah, I would say that's red. And what's on her arms? Tattoos, right? So let me ask you, if she has kids, do you think they're going to have that color hair? I don't think so either. I mean, I don't know for sure. I'm not trying to judge, and I wouldn't judge anyway. But it looks to me like that's hair color. If she has kids, do you think they're going to come out with tattoos? No, right? So one of the things you're going to learn about that you already know, but we're going to dig deeper. You're going to learn about things that are actually heritable, right? So not everything on you, not everything about you is heritable. Not everything that happens to your body will be passed along to your offspring. Some of it will. Obviously not tattoos and hair color. But yeah, so we'll talk about that. There are things that can happen to your body, to your cells while you're alive that can cause changes that can be passed down to your offspring, but for the most part, they don't. Even health, right? Like even if you work out, you're really jacked and big old muscles, that doesn't mean your kid's gonna come out and be muscular, right? So anyway, we'll talk about that. This is obviously a picture, I shouldn't say obviously, but we have a picture of a man and his daughter. I only know that because that's what the textbook said. And also the point that the old textbook was getting at there was that it's the man that determines the sex of the baby. Because a female, as you're going to learn, well, as you mentioned, as I mentioned earlier, and as you're really going to learn in this chapter, a female, generally speaking, is XX chromosomes. Males are generally XY. So when females create eggs, right, when they create their gametes, no matter what, unless there's something wrong, 100% of her eggs are going to have an X in them. For males, when they create their sperm, right, when they create their gametes, they go through meiosis. Half of them, 50% of them are going to have X, 50% of them are going to have Y. So that's why the male is going to determine the sex of the child. I don't remember what the textbook was getting at with this picture. Let me see. I wrote it down though. Your chances of inheriting and passing along a genetic disease can often be determined from your family history. Yeah, so that's all that is. That's a family. So in this chapter, you're also going to learn a little bit about pedigrees. So we can say, all right, if this person has this chance of having a disease, and they have a child with this person who has this chance of having that same disease, then what are the chances that the kids are going to have that disease? So that's the kind of stuff we're going to learn. It's a very relevant chapter, sort of like the last one. Well, I don't know, meiosis and mitosis. So a lot of the stuff you learned in the previous chapters are going to be relevant to this course. But genetics, that's very relevant to your everyday life, in my opinion. Just like the old textbook, the old textbook included something called Biology and Society. That was like the beginning of the chapter, and they had a little story about something that's relevant to everyday life. I'm not going to spend time talking about it, but it is in the PowerPoint if you want to look at it. And it basically talks about dog genetics. So if you're interested in that, take a look. If it inspires you, write an extra credit paper about it. Here we go. It looks long. Actually, it is a little bit long. So the way the textbooks break down, the old textbook breaks down this chapter's 3 main points. First, we're going to talk about genetics and heredity. Then, after I've told you all these things, and this is how genetics works, then I'm going to say, but it doesn't always work that way. And then I'm going to teach you the exceptions to the rule. And then finally, we're going to look at all this again from the point of view of like really looking at the chromosomes. Now, that being said, as I'm teaching you this, I am going to be talking about how this is relevant in the context of chromosomes. So hopefully by the time we get to that bullet point, we can really kind of get through it because hopefully by then I've already told you about it. So any questions about how this chapter is going to go? All right, the first word for attendance is Navy. Today is their 250th birthday. Navy is the first word for attendance. So of course, if you're online, there should be a screenshot where I can see this chapter overview with genetics and heredity highlighted. All right, there's no questions, let's talk about it. And what I love about the old textbook, one of the reasons I kept this one, especially this chapter. This chapter kind of tells it to you as like a story. So the person who figured out most of the stuff that we know about genetics, at least the big stuff, was this guy that I'm about to tell you about. So I'm presenting it as a story as opposed to facts, which I think is, I don't know, at least more entertaining. Hopefully it'll make it easier for you to learn. The first thing you need to know is the definition of heredity, and that is the transmission of traits from one generation to the next, which is funny because I haven't even told you what traits are yet. There is a technical definition for that, and we'll get to that eventually. But yeah, so you need to know that. As usual, I don't want you to memorize anything verbatim, usually. That includes this. So yeah, you should know the definition of heredity, but not like, I don't want you to just be able to regurgitate those words. You should be able to put them in your own words if you needed to. Or more accurately, when you're taking an exam, and you see a different version of it, you're like, oh yeah, that's the definition. So anyway, any questions about heredity? All right, so that brings us to the next term, and I'm probably not even gonna ask you this because there's just gonna be questions about this subject, but this is not the definition of the subject, excuse me. Genetics. So if heredity is a transmission of traits from one generation to the next, genetics is simply the study of heredity. So again, I'm probably not going to ask you the definition of genetics, but there will be a lot of questions about the stuff that you're about to learn. Again, a lot of it's going to be hypothetical stuff, like if this thing mates with this thing, what are the chances of this thing happening? Or if we got this offspring, then what must have been the parents? Things like that. Just for you to keep in the back of your mind as you're learning this. So the guy, like I said, the guy who learned most of this, figured most of this stuff out, his name is Gregor Mendel. I'm not going to ask you any questions about him, but again, I do like the way the old textbook tells us as a story, like, hey, let's figure out what Gregor Mendel figured out. So in the 1860s with his work, he argued that parents pass their unique genes to offspring, which we're going to learn about. I put a little star next to genes because that term didn't exist back then. He called them heritable traits. So he argued that parents pass their heritable traits off to offspring. And of course, that wasn't groundbreaking. No one knew exactly how it happened, but no, they weren't dumb. You could see if this person mated with this person. Hey, the kids kind of look like the parents, right? So that part wasn't groundbreaking. But he didn't know the word genes, but what he was referring to were genes. And he argued that those things were responsible for those inherited traits. So that was kind of new. Because nobody knew exactly how it was happening, right? No one knew how, when you put these two parents together, the children end up looking like the parents. So that part was new. He also argued that genes retain their individual identities generation after generation, no matter how they're mixed up or temporarily masked. And that's a big one. So you're going to learn a lot about that one. And that was definitely new. So, for example, how many of you have blue eyes, but your parents don't have blue eyes? Like either of your parents? Same here. I have blue eyes, both my parents have brown eyes. And that's a great example of that. And that was probably really weird to people back in the day. It's like, wait, how did that happen? Probably people got stoned or punished for that, I bet. But anyway, it's because that gene for the blue eyes, it's still there. It was still there. My mom still has that gene. My dad still has that gene. It's just that gene wasn't, the word is dominant. You'll learn that later. So because of that, their brown eye gene prevailed in them. And for me, in Sigmund, it did not. And you'll see why as we talk about it. Anyway, that's the introduction. So any questions about the introduction before we really dig in to how he figured all this stuff out? Okay. So he did all of his work in a garden called Abby's Garden or Abbey Garden. So let's talk about what he did here. He studied garden peas. I'm not going to ask you questions about what he studied, but like I said, the old textbook presents this as a story, so will I. And I guess this would be good for a little bit of critical thinking. Why did he study garden peas? Well, because they were easy to grow, right? So for example, you don't want something that's really finicky, so I don't know if any of you try to grow plants, but some of them are really hard to grow, hard to keep alive. Or imagine if he was trying to study redwood tree, right? Think about you'd have to wait hundreds of years for one tree to grow up. But anyway, yeah, they're easy to grow. And also they come in many readily distinguishable varieties, which is helpful when you're looking at genetics, right? Because you want to look at how I'm going to mix this color flower with this color flower and see what happens. I'm going to mix this color pea with that color P and see what happens, right? So that's very helpful for him to do that. So that first bullet point, not very important as far as the exam is concerned. It's just an introduction. But these next 2 bullet points are important. You definitely need to know these words because I'm going to use them a lot, and there's going to be questions that use them. So the first term is character. Character is a heritable feature that varies among individuals. So far, the one example I've given has been eye color. Eye color is a character. Trait, and that's a word I used previously when I defined heredity, but now I'm giving you the official definition, that is a variant of a character. So again, using the same example, if eye color is the character, then blue is a trait. Green is a trait. Brown is a trait. So definitely make sure you understand the definitions and the relationship between those 2 terms. They will be used a lot, character and trait. What was helpful for Mendel was that when he was studying these things, like every character he studied only had 2 traits. So it's either this or that. So it's really helpful. Like think about how hard that would be for humans if it wasn't like that. Like again, for eye color, there's different eye colors. Even if there's like different shades of brown and different shades of blue, right? Eye color is not good. Height is really bad. Imagine if every human was like, all right, you're either tall or short. You're either 4'5" or 6'5", right? Imagine if every human was like that. But it's not like that, right? So again, everything he did had 2 distinct traits, which again, later in the chapter, after I tell you all these things that you need to know about the basics of genetics, then, like I said, then it's like, oh, but that's not always the case. Let's talk about exceptions to the rules. And there's going to be a lot of them. And that's the biggest one, because a lot of things, I would probably say most things, don't come in 2 distinct traits, right? There's all kinds of different traits. Anyway, any questions about that side? All right. This is just telling you the story. As far as the exam is concerned, this isn't very important, but let's talk about it anyway. An important advantage of the pea plants is that he could strictly control the reproduction, right? So he could make sure that this plant was mating with this plant. And I'll just show you why. You can read what's on there, but it'll make more sense when I show you a picture. So he could cut off the male parts and leave the female parts. And he could take the male parts, like the pollen from the male parts, off of one plant and bring it to the female parts of another plant. So he was in control of which plants were mating with which. It's really important, right? Otherwise, you can't study genetics unless you can control which individual is mating with which individual. That's all they were getting at there. This is, again, not very important. It's just part of the story. The way he did it by hand. Again, he would cut off the male parts, use it like a brush, like you can see here. Put the pollen on the female parts of the other, blah, 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 blah. Not very important for this chapter, but again, it's in there, so I included it. Any questions about that? All right, here's some other terms you definitely need to know because I'm going to use them to describe things, right? I'm going to teach you, as I'm teaching you, these are terms I'm going to use. And in the exam, I'm going to use these terms. Like the first term, for example, is purebred. But I was about to say, I'm not going to ask you the definitions of them, but purebred, I might ask you the definition of them. But the point is, even if I don't ask you the definition of any of these words that are bolded, they're at least going to be used in a question to ask you something else. If you don't know what they mean, then you're not really going to understand what the question means, then therefore, you're not going to do good on the exam. So anyway, let's start with purebred. This guy created purebred varieties of plants. And what is a purebred? A purebred self-fertilizes to produce offspring that are identical to the parent. So in other words, and again, it's just we're thinking about one trait, the one we usually talk about is the flower color. It's either purple or white. So he bred and bred and bred, like he did generation after generation after generation until he made sure that all these plants are purebred white. There was no genes for anything else in those plants. And on the other hand, the purple ones, he did it so many times that he knew those are purebred purple plants. So that's what a purebred is. And again, that is one of the few that I might actually ask you the definition. So you need to know that a purebred self-fertilizes to produce offspring that are identical to the parent. You know, and again, don't memorize that verbatim. Understand what those words mean. That way in the exam, you know, if you get something that looks slightly different than that, but basically the same thing, you'll know to choose it. So what he did to study these genetics was to cross 2 different purebreds. right? So purebred, again, just using the flower color as an example, there were many other traits he studied, but he would cross a purebred white flower plant with a purebred purple flower plant, and the result of that was called a hybrid. That's what a hybrid is, at least in this context. Hybrid is an offspring that is from two purebred parents. That is what hybrid is in this context. Now, if you're talking about agriculture, I don't know, people growing different types of fruits, for example, that might be a different thing. But in this context, that's what a hybrid is. Of course, if you're in my environmental biology class, then hybrids would be a completely different thing. Then you're talking about a vehicle that runs on ice or internal combustion, but also electricity. So, yeah, depends on the context. Anyway, the act of creating that hybrid was called a genetic cross. So when you're mating, when you're forcing two plants to mate, call that a genetic cross. And again, that's a term that I will use as I'm teaching you and in the exams. The parents, in this particular case, were purebreds, but either way, even when you're not dealing with purebreds, which we don't even talk about, but even if you weren't dealing with purebreds, when you're describing it, all right, I crossbred this plant or this plant with this plant, that would be the P generation. P generation are the first that you're crossing. And of course, again, in this case, we're talking about pea plants, but this is how it is with any genetics. If you're studying, I don't know, frog genetics, if you took two frogs and mated them, that would be the P generation. And that would still be called a genetic cross. So those hybrids, again, that's when you cross two purebred, right? You do a genetic cross between two purebreds, get a hybrid. But that hybrid is also technically considered the F1 generation. So another way of saying that is when you cross the P generation, then you get the F1 generation. Even if you're not dealing with purebreds and hybrids. Even if it's just two non-purebred things crossing, again, those two non-purebred things would be the P generation, and their offspring would be the F1 generation. And then when you cross the F1 generation with each other, their offspring would be called the F2 generation. So make sure you're familiar with that. This will be very important terms. Again, not that I'm going to ask you what those mean, but because I'm going to use them a lot in these lectures, and because I'm going to use them in the questions. So you're definitely going to need to be very familiar, mostly with the P generation, F1 generation, and F2 generation. Because again, a lot of times the questions are going to be, all right, we crossed this with this, and the outcome was this, and then we crossed that with that. What are the chances of this happening? So it's going to be a lot of that kind of stuff. Also, some basic math. So if it's been a while since you've done some math, you might want to brush up on that. Well, we'll talk about it when we get there. Rules of probability. We'll talk about it when we get there. So any questions about this slide? All right. The next word for attendance is goat, because the Navy mascot is a goat. His name is Bill the Goat. I'm sure he's having a grand old time right now somewhere. Well, I'll tell you where on the next word. Anyway, any questions about this slide? Okay, let's talk about the law of segregation. And there will be questions about the law of segregation. Before I get into this, I want to remind you, we also talked last chapter about the laws of, well, I didn't even say law yet, but I did introduce you to something called independent assortment, right? And then later in this chapter, we're going to talk about it again, and I'm going to call it the law of independent assortment. When I was a student, these things always confused me. The concepts were fine. I understood, like, when I was studying the laws of segregation, I got the concepts. I got how it worked. When I studied the law of independent assortment, I understood how it worked. For some reason, what caught me up was the names. So, like, I was like, I know how that one works, but was that one called laws of segregation, or was that one called independent assortment? So try not to let that confuse you. If it does, let me know, and we can just try to find some way to help you remember. But I guess it's more important to realize or understand how it works than what it's called. But anyway, yeah, let's talk about it. Laws of segregation. So Mendel performed experiments that tracked the inheritance of characters, for example, flower color, that occurred as two distinct alternatives, right? Two different alternative traits, like we mentioned earlier. Everything he studied, every character he studied, had two traits. And again, using flower as a color as an example, was white or purple. There was no purplish white. There was no white, purple, and red. It was just white or purple. And the results led him to formulate several hypotheses. I give you those hypotheses in the next couple of slides, but not this next exact one. this is just a demonstration of what we were talking about, right? So all these different characters, flower color, flower position, seed color, seed shape, pod color, stem length. These are all these different characters he looked at. And you can see here that for each one of them, there's only two traits, right? It's either this or that, which is helpful when you're studying the basics of genetics. So here's another term you need to know. So remember when I said when you cross the P generation, right? So you take this purebred plant, you take this purebred plant, and you cross them, and we call that a genetic cross. That is still a genetic cross, but now we're going to get more specific, and it's called a monohybrid cross. So there's different types of genetic crosses. The ones we're starting to describe are called monohybrid crosses, which is basically just what I've described. I'm just giving you a name, right? So like I said, everything he looked at came in two distinct traits, this or that. That's a monohybrid cross. Well, he was just looking at one character, and there was only two different versions, right? Two different traits of that one character. That's a monohybrid cross. You're probably going to get very familiar with the monohybrid cross because you're going to do something that I'm teaching you about hopefully later today called Punnett square, where you do your own monohybrid crosses, and this is how you figure out the odds of this or the odds of that happening. And then an example, the same example I keep using, and the next slide is going to be a picture of the words that are on here. But if you cross a purebred purple flower plant with a purebred white flower plant, he found that in the F1 generation, all the offspring had purple flowers. And he did this thousands of times too. So it was well established that he knew without a doubt, all right, I've got these purebred purple plants, purebred white plants. I'm going to keep breeding them, and he did. And every time, 100% of the time, the results were always a purple flower offspring. So that leads to, right, that's the observation, if we're talking about the scientific method, which leads to the question, well, what happened to the gene for the white flower? Of course, he did again. They didn't know the word genes back then, but that had to have been the question that he was asking, right? What happened for the factor? What happened to the thing that is responsible for the white flower? And it would appear, if that's the only studying, like the only experiments he did, could just conclude, well, obviously, that gene for the white flower must have disappeared. Of course, we know that's not the case now, but at the time, that must have been the assumption. And again, had he left it there, that would have been it, right? And he would have had plenty of data to show that. Like, he could have said, I've bred thousands and thousands of these plants, and 100% of the time, they always came out with a purple flower. Therefore, I conclude that the white gene has disappeared. But he didn't do that. And we'll talk about what he actually did. But anyway, here's a visualization of what's on this slide. So again, you got a purebred purple flower, purebred white flower, you cross them, and then that F generation 100% were purple. The only thing I don't like about this picture, it's a little misleading because it's, trying to simplify the process. But you have to think, this kind of implies that he took one purple plant, one white plant, and he had one purple plant. But that's not the case. Again, he did this thousands of times, right? So he took thousands of purple flower plants and crossed them with thousands of white flower plants and had thousands of purple flowers as the offspring. Right? So again, at that point, if that's all he did, then you could logically conclude that the white gene had disappeared. But instead, he kept going. Instead, he took, again, I don't like this picture because there should be a lot more than just one flower, right? There's thousands of plants. So he would take these F1 and cross them with each other. And then he found out, again, this is a little bit oversimplified, so this kind of implies that he did it, and there was 4 plants as the offspring, but there weren't. There was thousands of them. But amongst those thousands of the offspring, that F2 generation, about 75% of them were purple, and about 25% of them were white. He always got that ratio. Granted, it always changed slightly. It wasn't exactly 3-to-1, but it happened enough, and the numbers were so large that that's the ratio we now use. So he had demonstrated that that white gene didn't disappear. It was there somewhere because it always came back in the F2 generation. And when it did, it almost always came back in 25% of the offspring. And that'll be big. That's the kind of math that you're going to be learning here shortly. Again, basics with fractions, ratios, things like that. And that's the ratio we're dealing with, at least at first. So any questions so far? All right, so again, like I just said, now it's in writing. You can see it up here. The gene for the white flower did not disappear. It was just hidden when the purple flower gene was present. So he was then able to figure out that those F1 genes, or those F1 plants, so not the purebred, but the resulting offspring of mating those purebreds, he knew then they had to have one gene for purple and one for white. So even though they were all purple, obviously some of them, or they had the gene for white, otherwise they couldn't produce white plants. But they did, once they were crossed. Those offspring, the grandkids, if you will, the grand plants, some of them, about 25% of them were white. So any questions about the slide? Okay. From those results, he formulated 4 hypotheses. I want to give myself a little star to remind myself of something. Oh yeah, and other results. So he did, again, this is very oversimplified. So he did more than what I just showed you. It got a little bit more complicated than that, but that's basically what he did. And from the results of those basic things and many other things that we didn't even talk about, but from all those results, he has these 4 hypotheses. First, there are alternative versions of genes that account for variations in inherited characters. You need to know that. Not necessarily verbatim, but just know, like, for any character you're looking at, there's different versions of the gene that's responsible for it. So even though we're not going to talk about this yet, but eye color, right? There's different eye colors. Well, there's one, I don't know if I want to say this. It's not necessarily true, but let's assume it is. Let's oversimplify it. Let's assume that there's one gene responsible for eye color, right? And as we know, since people have different eye colors, there's obviously different versions of those genes. And even though I'm going to formally tell you this later, I'm going to go ahead and mention it now because I mentioned it in the last chapter. What was I going to say with that? Characters, different versions. Yeah. So as you remember when you learned about homologous chromosomes, like the thing about them was they had the same genes in the same spot. So this is still kind of getting to that, right? So when we're talking about those alternative versions of genes, again, let's say that eye color, and I'm really oversimplifying and making things up, but let's say there's one gene located, or one gene for eye color, and it's located on, I don't know, the middle of chromosome number 5, right? That's how it would be for every human unless there was something wrong with their genome, right? Everybody would have that same gene at the same location on the same chromosome. But going back to what this number 1 is saying, even though it's the same gene and the same location, there are different versions of the gene. So, any questions about number 1? And again, that's a really long-winded way of saying, hey, there's different versions of genes, right? All right, there's a word for that. And if you've done the pre-lab, you're probably already aware of this, but this word is called an allele. Definitely need to know that. That is definitely a word that I'm going to use a lot, allele. There's different versions of genes. So again, when I'm oversimplifying this eye color thing here, you know, there's the blue allele, there's the brown allele, there's the green allele. Again, oversimplification, eye color is a little bit more complicated than that, but it's good visualization. So any questions about what an allele is? Okay, so that brings us to the second hypothesis. For each inherited character, an organism inherits 2 alleles, one from each parent. So this is you're probably more bored than usual, I hope, in a way. I mean, I don't want you to be bored, but hopefully you're already so familiar with this. You're like, hey, yeah, I knew this, I just never used these words to describe it. But yeah, hopefully you knew that, right? For anything we're talking about, again, let's oversimplify it and talk about eye color. You get a gene from your mom, you get a gene from your dad. Just like when we talked about meiosis, right? When you produce those eggs in your sperm, all those, hey, it's still there. I don't want to erase it. All those chromosomes line up, you know, mom, dad, dad, mom, whatever. They all line up in the middle of metaphase 1, right? So there's different homologous chromosomes, and once those separate, you know, when you're looking at your sperm or your egg, there's a 50% chance that they got that mom chromosome and 50% chance that they got that dad chromosome. Or for being specific, because we're talking about genes now, not just chromosomes. That little gene right there in the middle of chromosome number 5, right? There's a 50% chance that your sperm, any given sperm you look at, is going to come from your mom, and 50% chance that you know, any given sperm or egg that you're looking at came from your dad. So, any questions about number 2? All right, here's some other words you probably already know if you did the pre-lab. These are words you're definitely going to need to know for the exam. This part of the exam is going to have a lot of problem solving, and questions are going to include words like this: homozygous, Homozygous is an individual or an organism, whatever, that has two identical alleles. So that's an individual whose alleles are the same. And of course, again, let me say this. This is what Mendel was figuring out, right? These are his hypotheses. And this is an oversimplification. So let me go ahead and say this too. A better way of saying this, as I'm going to tell you officially later in the chapter, instead of saying an organism has two identical alleles, I think a better way of saying it is all their alleles are the same. You'll see what I mean by that later, because again, not everything comes in just two versions. Sometimes there's, you'll see. So instead of saying two identical alleles, I think it's better to just say 100% of the alleles are identical. So any questions about what homozygous means? Of course, hopefully that's easy for you to remember because that prefix homo, right, that means same. So what do you think the next one is? One, because you can probably get at what I'm saying with the words, but also you've done the pre-lab. So what's the next term you think I'm probably going to give you? If one is homozygous, what do you think the other one might be?
Heterozygous.
Yes, thank you. Heterozygous. And that's when they have different alleles. And again, this came from the textbook verbatim. I think instead of saying an organism with two different alleles, I think it would be better to say different alleles. Doesn't matter how many of them. If they have any alleles that aren't the same for any given gene, that's heterozygous. So homozygous, they have the same alleles. Heterozygous, they have different alleles. Hoping that's easy for you. And again, think homo means same, hetero means different, right? Mixed up. So any questions about this before the next slide, which is kind of like a dad joke meme? I didn't make it, I found it. Okay. So there's heterozygotes. It might be hard for you to read, but it says, just allele uneven. I love dad jokes to begin with, especially when they're biology related. Anyway, let's move on to the next slide. The third of his hypotheses. Well, the next word for attendance is going to be Philadelphia. Be shortened to Philly if you want. That is where the Navy was founded on this day 250 years ago. Anyway, his third hypothesis. If you have two different alleles, and again, these are his hypotheses. Later in this chapter, I'm going to tell you about some exceptions to these rules. But it's nice to give you the basics and then talk about how the basics aren't 100% correct. But if you have different alleles, right, different versions, so if you're heterozygous, then one of those alleles is going to be dominant and one's going to be recessive. You definitely need to use those terms, know those terms, and I'm going to use them a lot. So if it's dominant, well, it just dominates over the recessive one. So just again, we talk about the purple flower thing, right? That F1 generation, every single one of them, even though you couldn't see it, but every single one of them had that white allele, that white flower allele, but they were all purple flower because that purple flower, that allele, was dominant. And to explain, let me see, I won't overcomplicate this. When you really dig into the molecular biology, which we'll do later, but not even to this extent, when you really dig into it, there's a molecular reason. There's a reason that this is happening. And it's always different. But one of the reasons could be, like in this case, it could be that the white gene doesn't call for a white flower. It's just a broken gene, right? So the purple flower, that's what it's normal, right? That gene, if you have that allele, that purple allele, that means you just have a normal working gene that says, hey, produce purple. But if you have the white allele, it isn't, again, in this case anyway, it's always different, but in this case, it's like just a broken allele. So the gene was there, it was just broken. So it isn't that it said, hey, make a white flower. It was just didn't have any instructions on how to make a color. So you have one gene that's not saying anything about how to make a color, one allele, excuse me, and one allele that says, hey, make purple. Obviously, it's going to turn out purple. And of course, if you have two alleles that both are broken and don't say anything about making color, then they're just going to come out white. Anyway, so that's dominant recessive. Here's another thing that's going to be very important, because again, on the exam, there's going to be a lot of questions about, all right, if you mix this thing with that thing, the dominant is always an uppercase. So D, B, in this case, a P, right? Dominant is a P. Recessive is always a lowercase. Actually, P is the bad one. Let's do a different one. Q because those look different, right? Recessive is always lowercase. So that will be important, because I may say, I may give you a question where I'm like, all right, this thing is mating with this thing. What are the chances of the offspring having this? Or I might say, this thing is mating with this thing, right? So when you get it, this is what it's going to look like. It's usually not going to be a sentence. Well, it might sometimes. Sometimes it'll be written like this. Sometimes it might say, homozygous dominant, blah, 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 mixed with a heterozygous, blah, blah, blah. What are the chances that their offspring will be this? Or it might say, heterozygous, whatever, crossed with another heterozygous, whatever it is. What are the chances of the offspring being this? So yes, sometimes you might get a sentence like that. Other times it may just be this. Like that way I don't have to verbally say it or write it out. You can just see that's what the cross is. So any questions so far? Okay, now the fourth and final hypothesis of his. A sperm or egg carries only one allele for each inherited character. We'll just stop at that part first, right? So again, for any character, right? If we're talking about a character that is controlled by a single gene, there's only two versions of that gene, two alleles of that gene. For any sperm or egg you look at, if you need to find that gene, there's only going to be one allele, right? You're not going to have both of the alleles. As long as nothing went wrong, there's only going to be one of the alleles. And remember, all your somatic cells, which is a good time to remind you that, right? All your cells that are not sperm or egg, for each of them, you have two versions of each allele, right? Because you have two versions of each chromosome. So you have two alleles, like we've already mentioned earlier. But once you go through meiosis, they segregate. Right? Those two alleles segregate. Right there at the beginning of the first meiosis, right? We take those homologous chromosomes, we line them up in the middle, and we segregate them. And those homologous chromosomes have genes on them. So those genes have been segregated. And that is the law of segregation. So you need to know the law of segregation is basically the word that we give to meiosis. Meiosis 1, right? We've segregated those alleles. Or if you're looking at it in a different context, we've segregated those chromosomes. Those homologous chromosomes, they've been segregated. You need to know that. You also need to know, and this is the part where people usually get it wrong on the exam. I don't remember the exact wording of the question, but it's basically about this part right here. A sperm or egg only carries one allele. That's all you need to know, right? Sperm or an egg, a gamete, only carries one allele for any given gene. Because again, even though you've got two alleles in you, once you create a sperm or an egg, you're segregating them, right? You're cutting your genetic information in half. Of course, when they come back together in fertilization, those alleles, the pairs of alleles, can come back together. You already knew that. Now we're just repeating everything we repeated in the last chapter. We're just kind of talking about it in a different context. Same thing happening, but we're just looking at it from a different lens. So the next slide I'm going to show you is basically a visualization of what's on this slide. But are there any questions before I show you the visualization? All right, here we go. So this is what you've already seen. I've already shown you this picture, except now we've drawn the alleles on there, right? So previously, you just saw this picture without the alleles, right? We didn't write the alleles for you. Same here, we didn't show you the alleles. Now we're just writing the alleles. So we know this purebred purple flower has to be homozygous dominant, right? Because it's purebred, that's how we know that it has two of those big alleles. We know that the white, because that is a recessive allele, has to have two of the small alleles. And again, it was purebred, so we do it that way. So that thing's going to go through meiosis, right? And these things are going to segregate, but even though they're getting segregated, it's still the only thing that thing has to offer is a big P. So no matter what, even though they're getting segregated, 100% of their gametes are going to be big P. Same here. Even though we're going to segregate those little P's from each other, still the only thing it has to offer is a little P. So 100% of their gametes are going to have a little P, right? But then you cross these, the only possible outcome, unless something goes wrong, would have to be a heterozygous offspring, right? It would have to be. You get a big P, you mix it with a small p, the only thing you're left with, then it's going to be a big P, small p, right? 100% of the time, unless something goes wrong, then the offspring will be big P, little p. Which again, to remind you, this is homozygous dominant, homozygous recessive, that is heterozygous. Right, so now when that thing goes through meiosis and produces sperm or egg, both, again, just like they did here, they're gonna segregate, right? Because that's what happens in meiosis, when we segregate, when we separate those homologous chromosomes from each other. So in this case, when you look at their gametes, 50% of their gametes are gonna be big P, and 50% of their gametes are gonna be small p. And then when you cross those, that's what this is, that you're gonna get really familiar with in lab. This Punnett square, right? You can see the possible outcomes. You can draw it out and figure out the possible outcomes, right? So again, 50% of the sperm is gonna be big P, 50% of the sperm is gonna be small p. 50% of the eggs are gonna be big P, 50% of the eggs are gonna be small p. You can draw it out and like see, you put that comes together, you'll have a homozygous dominant. If these two come together, you'll have a heterozygous. If these two come together, you'll have heterozygous. And if these two come together, you'll have homozygous recessive. You will need to do that. Hopefully between now and then, I come up with some worksheets that maybe I can give you for extra credit. It's nice, it's good problem solving. And you're gonna get at least a couple of questions on the exam where you don't technically have to use a Punnett square, but it would be best if you did. It would make it a lot quicker and easier if you can visualize it. So hopefully I'll get you familiar with these Punnett squares. But anyway, again, back to what we're really trying to talk about at this specific moment is the law of segregation, right? So again, these alleles segregate from each other in meiosis. So any questions about this slide? All right, so here it is in writing, everything I just told you. That's a Punnett square is what I just showed you, right? The Punnett square highlights the 4 possible combination of gametes. And what I showed you was a heterozygous crossed with a heterozygous. But remember, for the exam, I might ask you to do a Punnett square that's homozygous dominant crossed with a homozygous recessive, or a homozygous recessive crossed with another homozygous recessive, or I don't know, a heterozygous crossed with a homozygous recessive. Right? So these are the kind of things between now and then that you need to familiarize yourself with. It's not too difficult as long as you don't overthink it. Anyway, so that's what the Punnett square highlights, right? It shows you the 4 possible combinations of gametes. And later, I'll give you some basic math instead of just so you draw it up and then you can say, oh yeah, 75% are going to be this, 25% are this. Or you could word that as a ratio. Yeah, 3 to 1, right? So we'll talk about that when we get there. Each square represents an equally probable product of fertilization. So again, like the chances of this happening are just as likely as this happening and this happening and this happening. Of course, if we're talking about the fact like, are they homozygous, heterozygous, or homozygous recessive? In this case, and we'll get there officially later. But you can see here, the chances of being heterozygous is actually more than anything else, right? Because there's like 1 in 4 chance to be homozygous dominant, 1 in 4 chance to be homozygous recessive, 2 in 4 chance that it's going to be heterozygous. And that's the kind of basic math that you'll be doing in this exam. So any questions about Punnett squares or monohybrid crosses? We're done talking about that. Okay, so that's perfect timing. As usual, as a reminder, the last word for attendance is going to be you ask me a question about something that I presented today, and we'll just be done 1 minute and a few seconds early. Have a great week. I have a lot of catching up to do on grades, so if you need me, call me or come to my office. If you send me an email, it might take a while before I can get to it. So I'm here for you, but it might be a little bit slow at first. Anybody come in late? I think I got everybody. Okay.